Hello and welcome to our last section, section 3.5 in our chapter 3 video lecture series on nucleic acids. These are our most important macromolecules for us to be able to continue life because they contain our cell's genetic blueprint and have instructions for its functioning. So our learning objectives here are going to be to describe nucleic acid structure and define the two types of nucleic acids, explain DNA structure and role, and explain RNA's structure and role. So first, just taking a look at both of these. What does DNA stand for? It's for our deoxyribonucleic acid, whereas RNA is for our ribonucleic acid. And in terms of our DNA, that is going to contain all of our genetic material. And within eukaryotes, we'll find the DNA within the nucleus. We also find it within chloroplasts and mitochondria. And in prokaryotes, so our bacteria and our archaea, the DNA does not have a nucleus. Instead, it's within an area we call the nucleoid region. And our DNA also helps to control all of our cellular activities, turning certain genes on and off. Whereas RNA's role is mostly involved in protein synthesis. We don't really want our DNA to leave the nucleus or the area of the nucleoid region. So instead, we're going to create this intermediary type of molecule known as RNA, and in our case it's messenger RNA or mRNA, so that it can go and communicate with the rest of the cell. And other types of RNA, which you may have heard of, are rRNA, tRNA, and microRNA, and they're all also involved in the protein synthesis and regulation process. We'll touch on those more as we continue our discussion here. So both DNA and RNA are comprised of our monomers known as nucleotides. You could see them pictured over here, and we refer to them as our nitrogenous bases. So how do our DNA and RNA come together? What does it consist of? Well, first we are going to have our sugar. So it is going to be deoxyribose in DNA, and that sugar will change to ribose within RNA. We'll always have our phosphate group attached, and this creates our backbone of those structures. And the last thing we are going to see attached to our sugar is a nitrogenous base. So in DNA, we will see our cytosine, and I'm going to go in order here of how I have them written, guanine, our adenine, and our thymine. Within RNA, we have very similar bases, but the difference is we won't have thymine in RNA, but instead use uracil. And we also classify these nitrogenous bases into two groups. We have our purines, which are going to consist of adenine and guanine, and these are two carbon rings structures, two carbon nitrogen rings, that is. And then our pyrimidines are going to be made up of a single carbon nitrogen ring, and they consist of cytosine, thymine, and uracil. Let's focus on DNA first. This is going to be a double helix structure where we see that DNA backbone that we talked about, meaning we've got our deoxyribose sugar here and the phosphate on the outside, and those nitrogenous bases are going to be found more in the interior. Now notice that they are facing one another, so they are going to create hydrogen bonds with one another in order to give more stability to our DNA structure. So we'll see that adenine will always bind with thymine, creating a double hydrogen bond, whereas cytosine will always bind with guanine, forming a triple hydrogen bond. So this is our stronger bond here. Now, when we talk about RNA in the next slide, we'll note the same components here, but we will use uracil instead of thymine. Thymine will not exist in RNA. The other thing to note about DNA structure is that it is anti-parallel, meaning the strands are running in opposite directions. So we have one end of this strand called the 5' carbon end, whereas let's say of that same strand, this would be the 3' carbon end, and the matching strand would have the opposite. So the 5' end would be here, and the 3' end would be over here. And just a note of a term that we're going to be using a lot throughout the next couple chapters is transcription. That's when DNA is going to be able to dictate the structure of mRNA. Essentially, we'll open up DNA and copy it and use the DNA information to create an mRNA temporary strand that we'll be using to move out of the nucleus. So let's discuss RNA. So much to go through here. RNA, instead of having that double helix look to it, is going to have a single strand, and it will contain the ribonucleotides. So like we said, we'll have adenine pairing with uracil and guanine pairing with cytosine. And just to reiterate the structure we talked about a couple slides ago, we'll have the ribose pentose sugar attached to the phosphate group to create the backbone, and then we'll have the nitrogenous bases along that strand. We do have 4 different types of RNA. I mentioned mRNA already, and this is carrying the message from DNA, essentially the genetic information from DNA as a temporary strand. And we read this as sets of 3 bases, which we call codons. So if you look at this image over here, this is our mRNA strand, and each of these little spaces or little divots are representing the bases. And notice how we've got 3 of them here in an area known as a ribosome together. So 3 of these nucleic acids, or 1 codon, is going to code for a amino acid. So I'll talk about how that works a bit more in a moment, but let's talk about these other forms of RNA. Like we said, this is a ribosome. So we call this the rRNA, which is going to have some enzymatic activity that will help to add a peptide bond. I'm actually going to add one more image here for this to be a little bit clearer. There we go. I like this image a little bit better because we can see the codons over here. And I'm going to actually introduce our next one so we can better explain the process here. We have transfer RNA that is going to be utilized to carry the correct amino acid for the protein synthesis to take place. And this whole process that we see pictured here is called translation, meaning that we are going to take this mRNA strand, put it into the ribosome, which, yes, it's already placed here, and then one by one, we're going to have tRNA come in with what we call an anticodon to match up to the codon. So remember we said like G and C are going to pair up, and A's and U's are going to pair up. So we take that correct anticodon, and it has an attached amino acid to it. So as it comes in, it attaches into the ribosome. Now notice we've got 3 different slots in here. One of them is going to be more of an entrance for the tRNA, and as that comes in, we are going to form a peptide bond between the amino acids. You can see that that has already taken place here. So this will shift into the middle position here for that peptide bond to form. And then as it continues to shift, we will then have the exit site. So the tRNA will leave after having dissociated from the amino acid so that we can keep this amino acid chain growing. And I highly recommend that you watch some of the animations that I have in a supplemental video folder. I'm just going to play a small clip here because I really think it helps to see this in as an animation to understand this process.
tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation.
So we really saw how that polypeptide chain gets created. So if I had a strand, oh, here we go. There we go. If I had a DNA strand that had AATTGCGC and we were going to create a complementary RNA strand for that, we would pair up the A's with U's, right? UU. The T's would pair up with A's, AA, and then we've got C, G, C, G. And so what we're doing is reading this mRNA strand to match up those tRNAs with the amino acids. Of course, we're going to have the anticodon to what's written on that mRNA strand. And this is called translation. If I didn't mention that before, I forgot if I did. And then the last thing I want to mention is microRNA. And this helps to regulate gene expression by interfering with the expression of certain mRNA messages. And the last thing I have for you here is a comparison between DNA and RNA. So I will let you read through that on your own. I've mentioned some of them throughout this video.